Hello everyone, my name is Poonam Shivaji Kamble. I am explaining about the central dogma of life. Central dogma is discovered by Francis Crick in 1957. Then what is meant by central dogma? Central dogma is a process in which genetic information flows from DNA to RNA to form a functional group of proteins. Is called as central dogma. Central dogma also illustrates that the flow of genetic information in cells and DNA replication to form uh, DNA replication to form the coding of RNA by the transcription process and the coding for the proteins by the translation process. Then the biopolymer consists of the biopolymers consist of RNA and DNA that are the intended into the that are classified into again three parts: general transfer, special transfer, and unknown transfer. General transfer occurs in all types of cells, and special transfer occurs only in exceptional case. And central dogma takes place in two different steps, that is the transcription step and then second is the translation step. In the transcription step, then the transfer or it is the process in which information is transferred from DNA to RNA by the enzyme RNA polymerase. When the DNA strand undergoes it, the process again is divided into three parts, that is the promoter, structural gene, and then terminator. When the DNA strand forms the RNA is called as a template strand, and another strand is called as coded strand. The transcription process consists of four types, that is the mRNA, mRNA is a messenger RNA, tRNA is a transfer RNA, sRNA is a sRNA is a synthesis RNA, then fourth is a rRNA, that is a ribosomal RNA. That all are important for the transcription process. Then second is a translation. Translation is a process in which ribosomes are in cytoplasm and in endoplasm synthesis the proteins after the transcription process ribosome is called as translation. And the translation steps again divided into three parts, that is the elongation, termination, and initiation. In the initiation step, it is stop in the initiation step, stop codon takes place, and in the second is the last. In the initiation step, the ribosomes assembles the target DNA, and the first stage are attached to the standard DNA. Then in the elongation, the last tRNA validated for the small ribosomal subunit. And the termination step, in that when stop codon is attached to the ribosome, then they realize the polypeptide, the ribosomal complex, and contain the muscle and contain the moves on the stop codon. And the next is a genetic code. Genetic code contains the information of the protein manufactured from RNA. There are basically three nucleotides and four nitrogen bases, which collectively form a triplet codon. That codon for a one amino acid from a triplet codon. That codes for one amino acid. Therefore, the number of possible amino acid range to 4 into 4 into 4 is equal to 64. of amino acids. There are 20 naturally existing amino acids. The genetic code degenerate. This was explained by the feature of the genetic code, according to which a few amino acids are coded by more than one codon, thus causing them to degenerate. Each codon codes for only use specific amino acid, and the codes are universal irrespective of the type of organism. Thank you.